Carbonara is an innovative AI-driven model developed by researchers at EPFL for predicting protein sequences from backbone scaffolds, while incorporating complex molecular environments. It represents a significant advancement in protein engineering by enabling context-aware sequence generation, which is particularly useful for integrating protein design with molecular environments, including non-protein entities. Carbonara leverages geometric deep learning through a transformer of atomic coordinates, trained by directly relating protein sequences, using only the atomic coordinates and element names of the atoms that make up the molecular systems. It builds upon the architecture of the protein structure transformer PESTO framework and uses geometric transformers, which are deep learning models processing spatial relationships between points, such as atomic coordinates, to learn and predict complex structures. The framework is trained on structural data available in the Protein Data Bank, PDB. All datasets, methods, and the Carbonara source code will be released upon publication. At its core, Carbonara processes atomic coordinates and element names to predict the most likely amino acids at each position in a protein sequence. It takes backbone scaffolds accompanied by non-protein molecules as inputs and generates a space of potential sequences that can be further constrained by specific functional or structural requirements. Carbonara offers an unprecedented level of flexibility and depth in protein design by considering the molecular context surrounding the protein of interest, meaning it can craft regions specialized for binding ions, substrates, nucleic acids, lipids, other proteins, etc. This is achieved because Carbonara's core, based on the PESTO model, allows it to incorporate any kind of non-protein molecules, including nucleic acids, lipids, ions, small ligands, cofactors, or other proteins, into the process of designing a new protein. Carbonara's central function lies in its ability to predict amino acid sequences directly from protein backbone structures, with a groundbreaking capacity to integrate the influence of the surrounding molecular environment. This ability manifests through a series of core functionalities that redefine the landscape of protein engineering. Foremost, Carbonara delivers unparalleled sequence prediction capabilities. Given a structural framework composed of atomic coordinates and element identities, the model generates the most probable amino acid sequence capable of conforming to that particular arrangement. This process transcends simple sequence generation. It involves a sophisticated analysis of spatial arrangements and interactions to ensure the predicted sequence is both structurally sound and functionally relevant within its intended biological context. Beyond mere prediction, Carbonara empowers researchers with the capacity for true de novo protein design and precise mutational engineering. This dual functionality allows for the creation of entirely new proteins tailored to specific tasks or the strategic modification of existing proteins to enhance or alter their inherent properties. The ability to generate novel proteins and tweak already known proteins allows Carbonara to create proteins that interact with certain molecules. A defining characteristic of Carbonara is its aptitude for context-aware sequence generation. The model transcends traditional methods by considering the presence and influence of surrounding molecules during sequence prediction. This encompassing view extends beyond simple protein-protein interactions to include nucleic acids, lipids, ions, and other ligands, mirroring the complex and dynamic environments in which proteins naturally exist. This function is able to increase prediction accuracy. Furthermore, Carbonara offers unparalleled customization for specific design objectives. The model's architecture allows for the incorporation of functional or structural goals, enabling the optimization of sequence identity, minimization of similarity, or achievement of low sequence similarity. This feature provides a flexibility that allows users to shape the sequence to fit their specific needs. Finally, Carbonara is capable of handling and designing proteins that contain non-protein components. This makes it particularly useful for designing proteins that interact with cofactors, ligands, or other non-protein molecules, mirroring natural proteins. Carbonara holds significant importance due to its innovative approach to protein design, which addresses critical challenges in biotechnology, medicine, and materials science. Its ability to accurately predict protein sequences while considering the complex molecular environments in which proteins function sets it apart from existing methods. Carbonara is capable of incorporating any kind of non-protein molecules, including nucleic acids, lipids, ions, small ligands, cofactors, or other proteins, into the process of designing a new protein. This is made possible because Carbonara's core, based on the pesto model, allows it to consider the molecular environment surrounding a protein natively for absolutely any kind of molecule type. By accounting for these restraints imposed by molecular environments, Carbonara can design proteins with specific functions, making it a valuable tool for enhancing future drug discovery campaigns. One of the grand challenges in protein engineering is designing proteins de novo, meaning from scratch, to tailor their properties for specific tasks. Carbonara addresses this challenge by enabling the creation of custom-designed enzymes that can facilitate rare or non-existent reactions in nature. This capability is particularly valuable in the pharmaceutical industry for synthesizing complex drug molecules and in environmental technology for breaking down pollutants or plastics more efficiently. Carbonara's flexibility and accuracy pave the way for industrial applications and scientific research, including biotechnology for crafting selective enzymes and protein reagents, synthesizing complex drug molecules in the pharmaceutical industry, and breaking down pollutants in environmental technology. Carbonara's experimental validation further underscores its importance. Researchers used Carbonara to design new variants of the TM1 beta-lactamase enzyme, involved in antimicrobial resistance. Some predicted sequences, differing significantly from the wild-type sequence, folded correctly and preserved some catalytic activity at high temperatures when the wild-type enzyme was already inactive. Moreover, Carbonara has potential applications in molecular evolution studies. This is because Carbonara can optimize the sequence prediction by considering not only interacting protein partners, but also nucleic acids, lipids, small molecules, and ions. The field of protein design has evolved significantly over the decades, transitioning from manual methods to sophisticated computational approaches. Early efforts in the 1970s and 1980s relied on manual sequence optimization, using known protein structures, sequence composition, amino acid charges, and desired structural geometry. These first designs are attributed to Gut, who designed a reduced version of bovine ribonuclease, and to Urey and colleagues, who designed elastin-like fibrous peptides. This initial era, termed manual protein design, involved constructing sequences that folded in water and membranes to adopt folded conformations. The advent of powerful computers, amino acid conformation libraries, and force fields in the 1990s spurred the development of structure-based computational protein design tools. This second wave focused on computational design guided by physicochemical principles. Researchers employed mathematical equations to define backbone conformations and sidechain repacking algorithms to design the sequence. A major milestone was the successful design of a completely de novo protein whose structure was fully verified. The 2000s marked the beginning of a third wave, where the expanding database of crystallographic structures enabled fragment-based and bioinformatically informed computational protein design. Modern de novo design involves generating new proteins with sequences unrelated to those in nature, based on physical principles of intermolecular forces. These designs often require specific rules derived from native structures in the Protein Data Bank (PDB), such as idealized alpha-beta fold proteins favoring certain beta-hairpin chirality. Today, protein design is one of the most important tools available for protein engineering, holding great promise for biomedicine and bioengineering. Despite the remarkable advancements in protein design, significant challenges persist in accurately predicting protein structures and functions. One of the major hurdles is the complexity of the energy landscape that governs protein folding. Predicting the precise three-dimensional structure of a protein from its amino acid sequence remains a difficult task, as proteins can adopt multiple conformations, and accurately modeling the balance of forces that stabilize the native state is computationally intensive. Even with accurate structural predictions, translating structure to function remains challenging, necessitating the development of innovative approaches that bridge the gap between structural data and functional behavior. Another notable challenge lies in accounting for the dynamic and heterogeneous environments in which proteins operate. Most computational design methods have historically treated proteins as isolated entities, neglecting the intricate interactions they undergo with other molecules, such as ligands, cofactors, and other proteins within cellular environments. Neglecting these contextual elements has led to discrepancies between design proteins and their intended behavior. The design of proteins that interact with non-protein molecules or function within specific cellular contexts requires a more comprehensive understanding of the interplay between structure, dynamics, and the surrounding environment. Furthermore, experimental validation remains an essential step in the protein design process. Designed proteins often fail to exhibit the desired stability or activity, necessitating iterative rounds of design, synthesis, and characterization to fine-tune their properties. Carbonara and similar advances in protein design are poised to have a transformative impact on the scientific community, fostering new research avenues and accelerating discoveries across multiple disciplines. By providing a more reliable and flexible method for designing proteins with specific functions, Carbonara lowers the barrier to entry for researchers seeking to explore protein engineering solutions. This will have multiple effects, including the design of more effective medications and vaccines, the creation of protein-based nanomaterials, antibodies, and the ability to engineer proteins for environmental cleanup. Moreover, the development of Carbonara contributes to a deeper understanding of the fundamental principles governing protein structure, function, and evolution. By enabling the design of proteins with novel folds and activities, Carbonara allows scientists to test hypotheses about the relationship between sequence, structure, and function, thus refining our knowledge of protein biochemistry and molecular biology. This feedback loop between design and discovery has the potential to revolutionize our understanding of the molecular world and to inspire the development of new theoretical frameworks for protein science. Further development and application of these tools promises the ability to design proteins that perform functions not found in nature, pushing the boundaries of synthetic biology and biotechnology. The impact of Carbonara extends beyond academic research, as it has the potential to accelerate the translation of basic scientific discoveries into real-world applications. By facilitating the design of proteins with enhanced stability, activity, and specificity, Carbonara can contribute to the development of new therapeutics, diagnostics, and industrial enzymes. This would enable scientists with no expertise in computational modeling to develop proteins for research and therapeutic or industrially relevant applications. The open sharing of protein design insights and strategies allows researchers to leverage existing knowledge and tools, accelerating the pace of scientific progress. Carbonara's core architecture revolves around a geometric deep learning approach, primarily leveraging a transformer network that directly processes atomic coordinates. This design is built upon the protein structure transformer PASTO framework. Instead of relying on traditional sequence-based or homology-based methods, Carbonara takes atomic coordinates and element names as its primary input, enabling it to directly learn relationships between the physical structure of a protein and its corresponding amino acid sequence. This approach allows for the incorporation of the entire molecular environment, including non-protein molecules, into the design process. The architecture consists of geometric transformers that process spatial relationships between points, in particular, atomic coordinates, to learn and predict complex structures. The transformer architecture is well suited for capturing long-range dependencies and contextual information within protein structures. The architecture utilizes self-attention mechanisms to capture long-range dependencies within the protein structure. The model is trained using a substantial dataset extracted from the Protein Data Bank, PDB. This dataset consists of protein structures alongside their associated sequences and, crucially, data regarding any bound molecules or surrounding environmental factors. The training process involves optimizing the model to predict the amino acid sequence from the structural data. Once trained, the model can then be used to design novel protein sequences that are compatible with a specified backbone structure and surrounding molecular environment. It builds upon the architecture of the protein structure transformer PESTO framework and uses geometric transformers, which are deep learning models that process spatial relationships between points, such as atomic coordinates, to learn and predict complex structures. This allows it to be more precise and to better capture the nuance of the context it is trying to predict. The output of the architecture provides the most likely amino acids at each position in a protein sequence that are compatible with the input data. This approach offers flexibility in protein design and allows for the incorporation of non-protein molecules. By combining geometric deep learning with transformer networks and extensive structural data, Carbonara's core architecture enables accurate and versatile protein sequence prediction, pushing the boundaries of protein design and engineering. Carbonara undergoes a comprehensive data training process that leverages a vast dataset derived from the Protein Data Bank, PDB. This dataset comprises approximately 370,000 protein subunits for training, complemented by roughly 100,000 subunits for validation and 70,000 for testing. Crucially, the data incorporates not only protein structures and sequences, but also the diverse molecular environments in which proteins function, including interactions with other proteins, nucleic acids, lipids, ions, small ligands, and cofactors. This inclusion of diverse molecular environments is crucial for Carbonara to generate context-aware sequence predictions. The training process involves feeding the model structural data, represented as atomic coordinates and element names, and optimizing it to predict the corresponding amino acid sequence. Unlike some other methods, Carbonara does not rely on extensive parameterizations of surface calculations. The model's ability to parse and process any molecular entity near the protein backbone eliminates the need for amino acid-specific parameterizations. During training, geometric transformer operations gradually process information from increasingly larger local neighborhoods, ranging from 8 to 64 nearest neighbors. These operations equivariantly encode geometric information from the vectorial state while representing invariant quantities of the geometry, allowing the model to effectively capture spatial relationships within the protein structure. Through this rigorous training regimen, Carbonara learns to associate specific structural features with particular amino acid sequences, while also accounting for the influence of the surrounding molecular environment. This enables Carbonara to accurately predict amino acid sequences for novel protein structures and to design proteins with tailored functions and properties. The fact that it does not solely focus on proteins, but rather on the whole molecular environment, is what allows for the model's predictive power and applicability in real-life, complex biological systems. Carbonara's functionality relies on a sophisticated combination of algorithms, primarily rooted in geometric deep learning and transformer networks. The cornerstone of its architecture is the use of geometric transformers. These algorithms are specifically designed to process data in a way that is invariant to rotations, translations, and reflections, essential for handling protein structures where spatial relationships are crucial. Geometric transformers likely involve techniques like spherical convolutions or graph neural networks to encode the relationships between atoms in a protein. They provide a robust way of representing and learning from three-dimensional data. A central component of Carbonara is the protein structure transformer PESTO, on top of which Carbonara is based. This transformer is a deep learning architecture known for its ability to handle sequential data and capture long-range dependencies. In the context of proteins, This translates to modeling the relationships between different parts of the protein structure, allowing the model to understand how changes in one region can affect other parts. The self-attention mechanisms inherent in transformers enable the model to focus on the most relevant parts of the input data when making predictions, improving accuracy and efficiency. These algorithms are what allow it to parse, recognize, and process any molecular entity near the protein backbone. Given the reliance on geometric information, algorithms for nearest neighbor search are also likely used extensively. These algorithms would efficiently identify the atoms or residues in the protein structure that are closest to a given point, allowing the model to focus on local interactions. This information is then used by the transformer network to make predictions about the amino acid sequence. Because it works on atomic coordinate data, it needs a way to connect the location of the atoms with the molecular makeup. Finally, optimization algorithms, such as stochastic gradient descent (SGD) or its variant CG-ATOM, play a critical role in training the model. These algorithms are used to update the model's parameters during training, guiding it to minimize the difference between its predictions and the actual amino acid sequences. These algorithms operate using only the coordinates, allowing them to incorporate any kind of non-protein molecule, including nucleic acids, lipids, ions, small ligands, cofactors, or other proteins, into the process of designing a new protein. Carbonara distinguishes itself through several unique features that collectively enhance its ability to tackle complex protein design challenges. A primary distinction lies in its context-aware sequence generation, enabling Carbonara to consider the influence of surrounding molecules when predicting protein sequences. By incorporating the surrounding molecular context, such as protein interacting partners and nucleic acids, Carbonara achieves higher sequence recovery rates. Another standout feature is its reliance on geometric transformers. By using atomic coordinates and element names as input, Carbonara eliminates the need for extensive parameterizations and is adaptable to various scenarios. This approach allows it to parse and process any molecular entity near the protein backbone, including proteins, small molecules, lipids, ions, and water molecules. By leveraging Pesto's architecture, Carbonara can perform protein sequence prediction conditioned by a specific non-protein molecular context. Carbonara also boasts a unique method for imprint sequence sampling. This technique provides diverse sequences while balancing design flexibility with high-confidence predictions. By offering a range of potential sequences, researchers can explore a broader design space and optimize proteins for specific properties or functions. The unique ability of Carbonara to optimize sequence prediction and to consider molecular restraints makes the tool very helpful for future drug discovery campaigns. Finally, Carbonara's ability to work solely with element names and atomic coordinates provides it with increased flexibility. This focus allows the model to include any biological assemblies from the RCSB PDB into its training dataset. All of these unique features enable Carbonara to accurately predict amino acid sequences for novel protein structures and to design proteins with tailored functions and properties. Carbonara necessitates specific data inputs to function effectively, primarily revolving around structural information represented through atomic coordinates and element names. The model takes as input the coordinates and elements of the backbone atoms C-alpha, C, N, O, and adds virtual C-beta atoms using ideal bond angles and lengths. Specifically, Carbonara requires a protein backbone scaffold, typically provided as a PDB protein databank file. The program automatically removes hydrogens and side chains from the scaffold. By default, the software uses all atoms in the input file for the prediction, such as ligands, lipids, ions, and water, though this behavior can be toggled. You can also specify whether to ignore hetatom, non-protein atoms, or water molecules in the structure. The coordinates are typically Cartesian. The geometry is described using distances and normalized relative displacement vectors between each atom. In essence, Carbonara leverages the spatial arrangement of atoms to predict the most probable amino acid sequence. This allows the model to incorporate the influence of the surrounding molecular environment into its predictions, making it a powerful tool for protein design. Despite its innovations, Carbonara, like any computational model, possesses certain technical limitations. A potential constraint lies in the quality and completeness of the input data. While Carbonara can leverage structural information from the Protein Data Bank, PDB, and other sources, the accuracy of its predictions is inherently dependent on the quality of the provided structural data. Errors or incompleteness in the input structure can propagate through the prediction process, leading to inaccurate or unreliable results. Furthermore, the resolution of the structure can also play a significant role. High-resolution structures generally lead to better predictions, while lower-resolution structures may introduce uncertainty. Another limitation stems from the computational cost associated with geometric deep learning. Processing large protein structures with complex molecular environments requires substantial computational resources, including memory and processing power. While efforts have been made to optimize the model's efficiency, the computational demands may still pose a barrier for researchers with limited access to high-performance computing infrastructure. This can be even further exacerbated by the inclusion of very big assemblies of protein structures. A crucial limitation is inherent in the black box nature of deep learning models. Understanding the precise reasoning behind the model's predictions can be difficult, hindering the ability to interpret and validate the results. This lack of transparency can also make it challenging to identify potential biases or limitations in the model's training data or architecture. Although the model's code will be released upon publication, the underlying calculations can be so complicated that the inner workings are obscure to anyone who didn't work on the software. Finally, while Carbonara can consider a wide range of molecular interactions, it may still struggle to accurately model certain complex or subtle effects, such as long-range electrostatic interactions or dynamic conformational changes. These effects can be difficult to capture with static structural data, potentially limiting the model's ability to design proteins with highly specific or dynamic functions. As such, like all AI models, it is not perfect and should be treated with caution when considering its conclusions. Carbonara enters a field of protein design with established competitors, most notably Protein MPNN and ESM-F1. These existing methods also aim to predict protein sequences from structural information, but Carbonara distinguishes itself through its unique capabilities. Protein MPNN, Protein Multipurpose Neural Network, is a prominent sequence design algorithm known for its speed and accuracy in predicting protein sequences. ESM-F1 is another competitive method that has demonstrated high performance in sequence recovery tasks. However, these alternatives can only track protein molecules, disregarding non-protein elements. In contrast, Carbonara can recognize other molecular entities and take them into account when calculating the position-specific scoring matrix. This capability allows Carbonara to achieve higher accuracy in the prediction of residues close to interfaces and to be more adaptable to different use cases. While Carbonara performs on par with Protein MPNN and ESM-F1 for sequence prediction of isolated proteins or protein complexes, it offers the additional advantage of handling protein designs that involve non-protein molecules. This capability significantly broadens the scope of applications in the field of protein design. Carbonara appears thus more flexible than the alternatives because it can intrinsically parse any kind of molecular system and can thus be used to drive sequence design. When comparing Carbonara to other protein design methods, several performance metrics are crucial. These metrics include sequence recovery, structural accuracy, measured by RMSD, root mean square deviation, and diversity of generated sequences. Sequence recovery refers to the percentage of correctly predicted amino acids in a design protein, with higher percentages indicating better performance. Carbonara performs on par with protein MPNN and ESM, if1 for sequence recovery in isolated proteins or protein complexes. This means that in these scenarios, Carbonara achieves a similar level of accuracy in predicting the correct amino acid sequence for a given protein structure. Structural accuracy, as assessed by RMSD, measures the deviation between the predicted and actual protein structure. A lower RMSD value indicates a higher degree of accuracy. The nonpolar loss might also be considered as a metric. Sequence diversity reflects the range of different sequences that a design method can generate. While high sequence recovery is essential, generating diverse sequences can be advantageous for exploring a broader range of potential protein functions. While ESM, if1 may outperform in terms of diversity, the best diversity compromises sequence recovery. Carbonara's advantage lies in its enhanced ability to handle non-protein molecules in the sequence prediction process, which allows it to take into account a larger range of molecular entities. This is especially useful in the prediction of residues close to interfaces. While specific performance metrics for this aspect of Carbonara are still being developed, its unique capability suggests a superior performance in designing proteins that interact with complex molecular environments. Carbonara offers several key advantages over existing protein design methods, primarily stemming from its context-aware approach and geometric deep learning architecture. Unlike alternative methods like protein MPNN that primarily focus on protein molecules, Carbonara can recognize and account for other molecular entities, including ligands, ions, nucleic acids, and lipids. This capability leads to higher accuracy in predicting residues close to interfaces and makes it more adaptable to different use cases. Specifically, Carbonara's unique ability to incorporate non-protein molecules enhances sequence recovery rates, particularly at protein interfaces. Studies have shown significant improvements in sequence recovery when molecular contexts, such as protein interacting partners, nucleic acids, ions, lipids, ligands, and glycans are included. In contrast, providing a non-native molecular context, such as a larger ion, decreases the sequence recovery rate. This highlights Carbonara's sensitivity to the specific molecular environment and its ability to optimize sequence prediction based on the surrounding context. Furthermore, Carbonara leverages the protein structure transformer (PESTO) framework, enabling it to work solely with element names and atomic coordinates, eliminating the need for extensive parameterizations and allowing for adaptation to various scenarios. The geometric transformer used by Carbonara processes atomic coordinates and element names to predict the most likely amino acids at each position in a protein sequence. By not relying on amino acid-specific parameterizations or pre-calculations, Carbonara is fast and flexible. Carbonara's ability to consider the molecular environment surrounding a protein natively for any kind of molecule type makes it more adaptable and functional than alternatives. Despite its innovative approach and promising results, Carbonara, like any computational model, has certain limitations that must be considered. One significant constraint lies in the quality and completeness of the input data. Carbonara relies heavily on the structural information available in the Protein Data Bank (PDB), which serves as its primary training dataset. While this database is extensive, it may not encompass all possible protein structures and molecular interactions, potentially limiting the model's ability to predict sequences for highly novel or underrepresented protein structures. Another limitation stems from the inherent complexity of protein folding and function. While Carbonara can consider various molecular contexts, including non-protein molecules, it may still struggle to capture all the nuances of protein behavior in vivo. Proteins often undergo dynamic conformational changes and interact with a multitude of molecules in complex cellular environments, which may not be fully represented in static structural data. This could lead to discrepancies between predicted sequences and their actual functionality in biological systems. Furthermore, like many deep learning models, Carbonara faces the challenge of interpretability. While it can generate sequences with high accuracy, understanding the precise reasoning behind its predictions can be difficult. This black box nature may limit its applicability in scenarios where a clear understanding of the design process is crucial, such as in regulatory approval processes for engineered proteins in medical applications. Additionally, while carbonara has shown promising results in experimental validations, including the design of thermostable and catalytically active enzymes, its performance may vary across different protein classes and functional requirements. As with any computational tool, experimental validation remains essential to confirm the functionality of design proteins, which can be time-consuming and resource-intensive. While there is no direct cost-effectiveness analysis available for carbonara specifically, we can draw insights from cost-effectiveness studies of related computational approaches in protein design and other fields to understand the potential economic implications of this technology. Computational protein design (CPD) methods, including approaches like carbonara, have the potential to significantly reduce the time and resources required for protein engineering compared to traditional experimental methods. By enabling rapid in silico screening and optimization of protein sequences, these tools can substantially decrease the number of experimental iterations needed to achieve desired protein properties. This efficiency gain translates to reduced labor costs, lower material expenses, and faster time to market for engineered proteins, potentially leading to significant cost savings in drug discovery, industrial enzyme development, and other biotechnology applications. However, the cost-effectiveness of computational approaches like carbonara must be weighed against their accuracy and the need for experimental validation. While these tools can generate promising candidates, laboratory testing remains essential to confirm the predicted properties and functionalities of design proteins. The overall cost-effectiveness thus depends on striking a balance between computational predictions and experimental validation, with the goal of minimizing false positives that could lead to wasted resources in downstream testing. It's important to note that the cost-effectiveness of computational protein design tools can vary depending on the specific application and the complexity of the design problem. For instance, in the pharmaceutical industry, even modest improvements in the success rate of protein drug candidates could lead to substantial cost savings, given the high costs associated with drug development and clinical trials. In contrast, for industrial enzyme engineering, the cost-effectiveness might be more closely tied to the ability to rapidly generate and screen large numbers of enzyme variants. As computational methods continue to improve in accuracy and capability, their cost-effectiveness is likely to increase. Future studies directly comparing the economic outcomes of projects using advanced tools like carbonara against traditional protein engineering approaches will be crucial for quantifying the true cost-effectiveness of these computational methods in various applications of protein design. Carbonara has been designed with user-friendliness in mind, making it accessible to researchers and practitioners in the field of protein design. The framework can be easily installed using common package managers like pip and conda, allowing for quick setup on most systems. Its Python-based interface enables seamless integration into existing workflows with straightforward methods for loading models and sampling sequences. The tool offers flexibility in its input requirements, accepting standard PDB files as input for protein structures. Users can specify various parameters to customize the design process, such as the number of sequences to sample, which parts of the structure to redesign, and which amino acids to include or exclude. This granular control allows researchers to tailor the design process to their specific needs without requiring extensive programming knowledge. Carbonara's documentation provides clear examples and use cases, including a quick start notebook that guides users through basic operations. The framework's ability to handle various molecular contexts, including non-protein entities, without requiring additional setup or parameterization, enhances its user-friendliness. This feature allows researchers to focus on their design goals rather than wrestling with complex software configurations. However, it's important to note that while Carbonara strives for user-friendliness, it still requires a solid understanding of protein structure and design principles to use effectively. The tool's advanced capabilities, such as context-aware sequence generation and imprint sequence sampling, may require some learning curve for users new to computational protein design. Nevertheless, the provided documentation and examples aim to flatten this curve, making Carbonara accessible to a wide range of researchers in the field of protein engineering and design. Carbonara's effectiveness has been demonstrated through rigorous experimental validations, particularly in the challenging domain of enzyme engineering. A notable example is the design and testing of variants of the TM1 beta-lactamase enzyme. Using the TM1 beta-lactamase backbone scaffold in complex with a beta-lactam substrate nitrosphin, Carbonara generated potential new sequences predicted to retain beta-lactamase activity. The model's context-aware approach proved crucial, as without the substrate context, key catalytic residues like S70 and R244 were not predicted with high confidence. However, when the substrate was included, these critical residues were correctly identified. To test the designed TM-like enzymes, researchers employed Carbonara's imprinting feature to generate 900 diverse sequences. These were then ranked using AlphaFold's predicted local distance difference test, PLDDT, in single-sequence mode, a metric shown to correlate with experimental success and foldability. The top 10 sequences with the highest PLDDT scores were selected for in vitro validation. Remarkably, all 10 variants expressed at high yields, indicating the model's ability to design stable, expressible proteins. The experimental validation extended beyond mere expression to functional analysis. The designed variants exhibited beta-lactamase activity, with some showing catalytic efficiency comparable to or even surpassing that of the wild-type TM1 enzyme. Importantly, the Carbonara-designed sequences incorporated mutations known to be relevant for substrate profile extension and stability enhancement in TM beta-lactamases. These included substitutions like R164H, A237T, M182T, and R275Q, which are recognized in the field for their roles in expanding substrate specificity and stabilizing the enzyme structure. Furthermore, the study compared Carbonara's residuized amino acid probabilities with experimentally determined tolerances to amino acid substitutions in TM beta-lactamases. This comparison, based on deep sequencing data from a library of all single residue mutants, showed a strong correlation between the model's predictions and experimental results. This validation not only confirms Carbonara's accuracy in predicting viable sequences, but also highlights its potential as a tool for studying protein evolution and guiding targeted protein engineering efforts. These experimental validations underscore Carbonara's capability to design functional proteins with specific properties, demonstrating its potential as a powerful tool in the field of protein engineering and therapeutic development. The successful design and validation of enzyme variants with improved or altered properties showcase the model's ability to navigate the complex landscape of protein sequence structure-function relationships, opening new avenues for rational protein design and engineering. Carbonara has demonstrated significant potential in the field of enzyme engineering, particularly in designing thermostable and catalytically active enzymes. A notable example of its application is the engineering of variants of the TM1 beta-lactamase enzyme, which plays a crucial role in antimicrobial resistance. Using the TM1 beta-lactamase backbone scaffold in complex with a beta-lactam substrate nitrosphin, Carbonara generated potential new sequences predicted to retain beta-lactamase activity. The model's context-aware approach proved crucial in this process. When the substrate was included in the design context, critical catalytic residues like S70 and R244 were correctly identified with high confidence. The researchers employed Carbonara's imprinting feature to generate 900 diverse sequences, which were then ranked using AlphaFold's predicted local distance difference test, PLDDT, in single-sequence mode. The top 10 sequences with the highest PLD/T scores were selected for in vitro validation. Remarkably, all 10 variants expressed at high yields, indicating the model's ability to design stable, expressible proteins. Experimental validation revealed that some of the Carbonara-designed sequences differed by approximately 50% from the wild-type sequence, yet still folded correctly and preserved catalytic activity at high temperatures where the wild-type enzyme was already inactive. This achievement demonstrates Carbonara's capability to design highly thermostable, catalytically active enzymes, which has significant implications for various applications. The success in designing thermostable beta-lactamases is particularly noteworthy, as these enzymes have potential applications in the inactivation of beta-lactam antibiotics from wastewaters. More broadly, thermostabilized proteins have substantial market value in several manufacturing and biotechnological industries. Carbonara's approach to enzyme engineering represents a significant advancement in the field of computational protein design. By incorporating complex molecular environments and leveraging geometric transformers to process atomic coordinates, Carbonara offers a more nuanced and context-aware method for enzyme design. This capability allows for the creation of enzymes with enhanced stability, altered specificity, or improved activity, opening new avenues for industrial applications and scientific research in fields ranging from pharmaceuticals to environmental technology. Carbonara represents a significant advancement in the field of drug discovery and design, offering a powerful tool for developing novel therapeutic proteins and optimizing existing ones. By leveraging its context-aware approach and ability to incorporate complex molecular environments, Carbonara addresses several key challenges in the pharmaceutical industry, particularly in the early stages of drug development. One of Carbonara's most promising applications is in the design of therapeutic proteins with enhanced developability profiles. The model's ability to predict amino acid sequences while considering the surrounding molecular context allows for the creation of proteins that are not only functionally effective, but also stable and less prone to aggregation. This is crucial in the pharmaceutical industry, where the complex nature of protein-based therapeutics often leads to challenges in formulation and clinical application. Carbonara's approach could potentially reduce the high attrition rates and costs associated with protein drug development by identifying optimal sequences that balance efficacy with favorable physicochemical properties. In the realm of de novo protein design, Carbonara opens up new possibilities for creating novel therapeutic proteins that do not exist in nature. By exploring a vast sequence space beyond naturally occurring proteins, the model can generate candidates with desired structures and functions, such as high-affinity binding to specific targets. This capability is particularly valuable in developing new classes of protein-based drugs, including engineered antibodies, cytokine mimics, and other biologic therapies. The success of de novo design proteins like NL-201, an IL-2 mimic with reduced toxicity and enhanced anti-tumor effects, demonstrates the potential of this approach in creating more effective and safer therapeutics. Furthermore, Carbonara's experimental validation in enzyme engineering, particularly with the TM1 beta-lactamase variants, showcases its potential in designing proteins with improved stability and catalytic activity. This has significant implications for developing new antimicrobial agents and other enzyme-based therapies. The model's ability to identify mutations that enhance substrate specificity and stability while maintaining function could lead to the development of more effective and resilient therapeutic enzymes. As computational methods continue to advance, tools like Carbonara are poised to transform the drug discovery and design process, potentially accelerating the development of new therapeutics and reducing the associated costs and risks. By providing a more rational and efficient approach to protein engineering, Carbonara and similar technologies could democratize drug discovery, enabling smaller research groups and companies to contribute to the development of novel protein-based treatments. This shift towards computational-driven drug design promises to expand the repertoire of therapeutic options available to patients across a wide range of diseases. Carbonara's success in enzyme engineering, particularly in designing thermostable and catalytically active enzymes, has opened up significant opportunities in industrial biotechnology. Thermostable enzymes, which can withstand elevated temperatures without altering their structure or function, offer enormous potential as biocatalysts across various industrial sectors. One of the most promising applications of Carbonara-designed enzymes is in the field of biofuel and biomass production. Thermostable enzymes like cellulases and xylenases have garnered considerable interest for their ability to efficiently break down complex lignocellulose polymers into fermentable monosaccharides. This process is crucial for the production of biofuels and other bio-based products. The use of thermostable enzymes in these applications offers several advantages, including increased reaction rates, reduced risk of microbial contamination, and improved substrate solubility. Carbonara's ability to design highly thermostable, catalytically active enzymes has been demonstrated through its work with beta-lactamase variants. These engineered enzymes, which retain activity at high temperatures where wild-type enzymes are inactive, have potential applications in wastewater treatment, specifically in the inactivation of beta-lactam antibiotics. This application addresses growing concerns about antibiotic resistance and environmental contamination. The food and beverage industry is another sector that stands to benefit from Carbonara-designed thermostable enzymes. Enzymes like alpha-amylases are widely used in this industry, and thermostable variants offer improved efficiency and stability in processes such as starch liquefaction and saccharification. Similarly, in the textile industry, thermostable lactases designed by Carbonara could find applications in fabric treatment and dye decolorization. The pharmaceutical industry also stands to benefit from Carbonara's capabilities. Thermostable enzymes are valuable in drug synthesis processes, offering increased reaction rates and reduced risk of contamination. Furthermore, the ability of Carbonara to design enzymes that function in complex molecular environments makes it particularly suited for developing biocatalysts for challenging pharmaceutical synthesis reactions. As industrial processes continue to move towards more sustainable and environmentally friendly practices, the demand for efficient biocatalysts is likely to grow. Carbonara's ability to design thermostable enzymes with tailored properties positions it as a valuable tool in this transition, potentially revolutionizing various industrial biotechnology applications. Carbonara's potential to revolutionize the field of medicine is significant, with applications ranging from drug discovery to diagnostic tools. This AI model for protein engineering promises advancements across various medical fields, including biotechnology. One of the most exciting prospects is in the realm of cancer treatment and prevention. A groundbreaking discovery involving graphene oxide, a material related to the field of protein engineering, has shown promise in neutralizing cancer stem cells. While not directly linked to Carbonara, this breakthrough illustrates the potential for novel materials and approaches in cancer treatment. Researchers found that graphene oxide flakes could prevent cancer stem cells from forming tumor spheres, effectively forcing them to change into non-cancer stem cells. This selective neutralization of cancer's root, while remaining harmless to healthy cells, represents a significant step forward in cancer research. Although this specific breakthrough predates Carbonara, it highlights the potential for innovative approaches in protein engineering to address complex medical challenges. In the realm of diagnostics, Carbonara's ability to design proteins with specific properties could lead to the development of more accurate and sensitive diagnostic tools. An example of innovation in this area is the AlgameterX device, which objectively identifies pain type and intensity using digital images of a patient's pupillary response. While not directly related to Carbonara, this technology demonstrates the medical field's move towards more objective and precise diagnostic tools. Carbonara could potentially contribute to the development of similar advanced diagnostic proteins or enzymes. The potential of carbonara in designing thermostable enzymes also has significant implications for medical applications. Thermostable enzymes are valuable in drug synthesis processes, offering increased reaction rates and reduced risk of contamination. These enzymes could lead to more efficient and cost-effective production of pharmaceuticals. Furthermore, the ability of carbonara to design enzymes that function in complex molecular environments makes it particularly suited for developing biocatalysts for challenging pharmaceutical synthesis reactions. As we look to the future, the potential medical breakthroughs enabled by carbonara are vast. From developing new therapeutic proteins with enhanced stability and efficacy to creating novel diagnostic tools and improving drug manufacturing processes, this AI model for protein engineering stands poised to make significant contributions to medical science. However, it's important to note that while the potential is exciting, the path from computational design to clinical application is long and rigorous. As with any medical innovation, extensive testing and clinical trials will be necessary to ensure the safety and efficacy of carbonara-designed proteins before they can be widely used in medical treatments. Carbonara's innovative approach to protein design has significant implications for molecular evolution research. By incorporating complex molecular environments and leveraging geometric transformers to process atomic coordinates, carbonara offers a more nuanced understanding of protein evolution and the relationship between sequence, structure, and function. One of the key impacts of carbonara on molecular evolution research is its ability to provide insights into the evolutionary constraints imposed by protein structure. Traditional models of protein-coding gene evolution often neglect protein structure, despite evidence that it plays a crucial role in shaping evolutionary patterns. Carbonara's context-aware approach allows researchers to explore how different molecular environments influence protein evolution, potentially revealing new insights into the forces driving molecular adaptation. The model's ability to improve sequence recovery, especially in cases with previously low recovery rates, by applying it to structural trajectories from molecular dynamic simulations, is particularly noteworthy. This capability opens up new avenues for studying protein evolution under different environmental conditions and conformational states. It could help researchers better understand how proteins adapt to new functions while maintaining structural integrity, a central topic in protein evolution. Furthermore, carbonara's ability to study the effect of specific contexts locally as well as their long-range influences on amino acid predictions hints at its potential for investigating evolutionary trade-offs and constraints. This could provide valuable insights into how proteins evolve to balance multiple, sometimes conflicting, functional requirements. The integration of carbonara with other evolutionary models and tools could lead to more comprehensive approaches for studying molecular evolution. For instance, combining carbonara's structural insights with phylogenetic analyses could enhance our understanding of how protein structure influences evolutionary rates across different lineages. This could potentially refine existing models of sequence change that incorporate effects on evolutionary rates due to protein tertiary structure. As of February 2025, while carbonara has shown promise in protein design and engineering, its full potential in molecular evolution research is still being explored. However, its unique capabilities suggest that it could become a valuable tool for evolutionary biologists seeking to unravel the complex relationships between protein sequence, structure, and evolution. As more researchers adopt and adapt this technology, we can expect to see a wealth of new insights into the molecular mechanisms of evolution in the coming years. Carbonara's potential in agricultural applications stems from its ability to design thermostable enzymes with enhanced properties, which could revolutionize various aspects of agricultural production and processing. While carbonara itself has not been directly applied in agriculture, as of February 2025, its capabilities in enzyme engineering suggest promising future applications in this field. Thermostable enzymes have significant advantages in agricultural processes, particularly in the production of biofuels and the treatment of agricultural waste. For instance, thermostable cellulases and xylenases, which Carbonara could potentially design, are crucial for the efficient breakdown of lignocellulosic biomass into fermentable sugars for biofuel production. These enzymes' ability to function at high temperatures increases reaction rates, reduces the risk of microbial contamination, and improves substrate solubility, making the overall process more efficient and cost-effective. In the realm of crop protection and pest management, Carbonara-designed thermostable enzymes could play a vital role. For example, thermostable lipases and proteases could be developed for use in biopesticides, offering more stable and long-lasting formulations that can withstand the harsh environmental conditions often encountered in agricultural settings. These enzymes could provide more environmentally friendly alternatives to traditional chemical pesticides, aligning with the growing demand for sustainable agricultural practices. Furthermore, Carbonara's potential in designing enzymes that function in complex molecular environments could lead to the development of more efficient fertilizers and soil amendments. Engineered enzymes could enhance nutrient availability in soils, improve plant nutrient uptake, or even help in the biodegradation of persistent organic pollutants in agricultural soils. This could contribute to more sustainable farming practices by reducing the reliance on chemical fertilizers and improving soil health. In the food processing sector of agriculture, thermostable enzymes designed by Carbonara could find numerous applications. For instance, thermostable amylases could be used in the production of syrups from starch, while thermostable proteases could be employed in cheese production or meat tenderizing processes. These enzymes' ability to withstand high temperatures could lead to more efficient processing methods, reduced energy consumption, and improved product quality. As agriculture faces increasing challenges from climate change and the need for sustainable practices, the potential of Carbonara in designing tailored, thermostable enzymes offers exciting possibilities. From enhancing biofuel production to improving crop protection and food processing, Carbonara-designed enzymes could play a crucial role in shaping the future of agriculture. However, it's important to note that as of early 2025, these applications are still largely theoretical and would require extensive research, development, and regulatory approval before being implemented in real-world agricultural settings. While Carbonara was primarily developed for protein design and engineering, its potential applications in materials science are intriguing, albeit largely theoretical at this point. The model's ability to work with atomic coordinates and element names without requiring extensive parameterizations suggests it could be adapted for materials science applications, particularly in the design and optimization of novel materials at the molecular level. One potential application of Carbonara in materials science could be in the field of biomaterials. The model's ability to design proteins with specific properties could be leveraged to create new biomaterials with tailored characteristics for medical implants, tissue engineering scaffolds, or drug delivery systems. By incorporating the molecular context of the surrounding environment, Carbonara could potentially design proteins that interface effectively with synthetic materials, improving biocompatibility and functionality. Another promising area for Carbonara's application in materials science could be in the development of bio-inspired materials. The model's success in designing thermostable enzymes suggests it could be used to create proteins that mimic or enhance the properties of natural materials. For instance, it might be possible to design proteins that contribute to the strength and flexibility of synthetic materials inspired by natural structures like spider silk or nacre. In the realm of nanotechnology, Carbonara's ability to work with complex molecular environments could be valuable for designing protein-based nanostructures or nanodevices. By predicting protein sequences that can self-assemble into specific structures or interact with other nanomaterials, the model could contribute to the development of new nanomaterials with applications in electronics, energy storage, or environmental remediation. However, it's important to note that as of February 2025, carbonara's direct applications in materials science remain largely unexplored. The model's potential in this field is based on extrapolation from its capabilities in protein design and engineering. Significant research and development would be required to adapt carbonara for materials science applications, including training on relevant datasets and validating its predictions in experimental settings. As the field of AI-driven materials design continues to evolve, models like carbonara may play an increasingly important role in accelerating the discovery and optimization of new materials. As of February 2025, carbonara has already demonstrated significant potential in protein design and engineering. However, several planned developments are on the horizon to further enhance its capabilities and expand its applications. One of the key areas of focus is improving carbonara's ability to design proteins with enhanced stability and functionality. The successful design of TM1 beta-lactamase variants that retain catalytic activity at high temperatures has opened up new possibilities for enzyme engineering. Researchers are likely to build on this success by developing carbonara's capabilities to design thermostable enzymes for various industrial applications, including biofuel production, waste treatment, and pharmaceutical synthesis. Another planned development involves expanding carbonara's training dataset. Currently, the model is trained on approximately 370,000 subunits from the Protein Data Bank, PDB, with additional data for validation and testing. Future iterations of carbonara may incorporate larger and more diverse datasets, potentially including data from other structural databases or even computationally predicted structures. This expansion could enhance the model's ability to handle a wider range of protein structures and molecular environments. Researchers are also likely to focus on improving carbonara's context awareness capabilities. The model's unique ability to consider complex molecular environments in its predictions has proven valuable for designing proteins that function in specific contexts. Future developments may aim to refine this feature, allowing for even more precise predictions of protein sequences in complex biological systems. Integration with other computational tools and experimental techniques is another area of planned development. By combining carbonara with other AI models, molecular dynamic simulations, or high-throughput experimental methods, researchers could create more comprehensive protein design pipelines. This integration could lead to faster and more accurate protein engineering processes, accelerating the development of novel therapeutics and industrial enzymes. As carbonara continues to evolve, its potential applications in fields such as medicine, biotechnology, and materials science are likely to expand. Researchers may focus on adapting the model for specific challenges in these fields, such as designing proteins for targeted drug delivery, creating novel biomaterials, or engineering enzymes for environmental remediation. These developments could significantly impact various industries and contribute to solving pressing global challenges. As carbonara continues to evolve, its potential for new applications extends beyond its current focus on protein design and engineering. While the model was initially developed for predicting protein structures, its unique capabilities suggest promising avenues for expansion into diverse fields. One potential new application lies in the realm of climate technology and carbon dioxide removal (CDR). As the urgency to address climate change intensifies, carbonara's ability to design thermostable enzymes could be leveraged to develop more efficient catalysts for carbon capture and utilization processes. For instance, the model could potentially be adapted to design enzymes that can operate effectively in the harsh conditions often encountered in industrial carbon capture settings. This application could contribute significantly to scaling up CDR technologies, which are becoming increasingly crucial in the fight against climate change. In the agricultural sector, carbonara's potential for designing enzymes that function in complex molecular environments could lead to innovations in sustainable farming practices. The model could be used to develop enzymes for enhancing soil carbon sequestration or improving the efficiency of biochar production. These applications align with the growing interest in nature-based solutions for carbon removal and could help address challenges in scaling up agricultural CDR strategies. The pharmaceutical industry might also benefit from new applications of carbonara. Its ability to design proteins with specific properties could be extended to the development of novel drug delivery systems or the creation of more stable and effective biopharmaceuticals. This could potentially accelerate the drug discovery process and lead to more targeted therapies. In materials science, carbonara's approach to protein design could be adapted to create bio-inspired materials with unique properties. For example, the model could be used to design proteins that self-assemble into specific nanostructures, opening up possibilities in fields such as nanotechnology and advanced materials engineering. As we look towards 2025 and beyond, the potential for new applications of carbonara seems vast. However, it's important to note that realizing these possibilities will require continued research, development, and validation. The model's adaptability to different molecular contexts suggests that its impact could extend far beyond its original purpose, potentially contributing to solutions for some of the most pressing challenges in science, technology, and environmental sustainability. Carbonara represents a significant advancement in the field of biotechnology, offering unprecedented capabilities in protein design and engineering. This AI-driven model's ability to predict protein sequences while considering complex molecular environments has far-reaching implications for various biotechnological applications. One of the most promising aspects of carbonara is its potential in enzyme engineering. The model has demonstrated success in designing highly thermostable, catalytically active enzymes, as evidenced by its work with TM1 beta-lactamase variants. These engineered enzymes, which retain activity at high temperatures where wild-type enzymes are inactive, have potential applications in industrial processes and environmental remediation. For instance, thermostabilized lactamases could be used for the inactivation of beta-lactam antibiotics in wastewaters, addressing growing concerns about antibiotic resistance and environmental contamination. Carbonara's flexibility and accuracy open new avenues for protein engineering in biotechnology. Its unique ability to take into account complex molecular environments makes it a valuable tool for designing proteins with specific functions, potentially enhancing future drug discovery campaigns. This capability could lead to the development of more effective and targeted protein-based therapeutics, offering alternatives to traditional small molecule drugs. In the realm of industrial biotechnology, carbonara's ability to design thermostable enzymes could revolutionize various processes. Custom-designed enzymes can facilitate rare or non-existent reactions in nature, which is particularly valuable in the pharmaceutical industry for synthesizing complex drug molecules and in environmental technology for breaking down pollutants or plastics more efficiently. Furthermore, carbonara's approach to protein design could contribute to advancements in synthetic biology. By enabling the design of proteins with novel functions or improved properties, it could help in creating new biological systems or enhancing existing ones for biotechnological applications. As biotechnology continues to evolve, tools like carbonara are poised to play a crucial role in accelerating innovation and discovery. From developing new biocatalysts for industrial processes to creating novel protein-based materials, carbonara's context-aware approach to protein design offers exciting possibilities for pushing the boundaries of what's possible in biotechnology. The rapid advancement of AI-driven protein design technologies like carbonara has brought ethical and regulatory considerations to the forefront of scientific discourse. In March 2024, over 100 senior scientists from more than 20 countries signed a statement outlining community values, guiding principles, and commitments for the responsible development of AI for protein design. This initiative, which emerged from a summit on AI safety convened by the UW-Medicine Institute for Protein Design, underscores the scientific community's recognition of both the immense potential and the possible risks associated with these technologies. The statement emphasizes that while The benefits of current AI technologies for protein design far outweigh the potential for harm. A proactive risk management approach is necessary to mitigate the risk of misuse. Key values articulated in the statement include safety, security, equity, international collaboration, openness, responsibility, and pursuing research for the benefit of society. These principles aim to guide the development of AI in protein design to ensure it remains beneficial for all. One of the primary ethical considerations is the dual-use nature of protein design technologies. While they have the potential to create new vaccines, medicines, and sustainable materials, there is also a risk that they could be misused to produce harmful substances or bioweapons. To address this, the scientific community has committed to participating in and improving the process of nucleic acid synthesis screening, aiming to detect hazardous biomolecules before they can be manufactured. Regulatory considerations are also crucial in this rapidly evolving field. The statement calls for collaboration with governments, civil society, funders of science, academics, and the private sector to ensure that research benefits society. This includes working on applications that promote health, sustainability, security, and scientific knowledge, while refraining from those likely to result in harm. The signatories also emphasize the importance of engaging stakeholders with diverse perspectives and informing the public about the benefits, limitations, risks, and risk mitigation strategies of their work. Transparency and openness in research are highlighted as essential principles, with the caveat that access to AI systems should be limited only when they present identified meaningful and unresolved risks. This approach aims to balance the need for scientific progress with the imperative of safety and security. The statement also stresses the importance of continuous assessment of protein design software, including how these tools intersect with other technologies, to apply appropriate safety and security measures. As the field of AI-driven protein design continues to advance, these ethical and regulatory considerations will likely evolve. The scientific community's proactive approach in addressing these issues demonstrates a commitment to responsible innovation. However, ongoing dialogue and collaboration between scientists, policymakers, and the public will be crucial to navigate the complex ethical landscape and develop appropriate regulatory frameworks for this transformative technology. As we look towards the future of AI-driven protein design, Carbonara stands at the forefront of a rapidly evolving field. The next frontier for this technology extends beyond its current applications, promising transformative impacts across various sectors. One of the most exciting developments on the horizon is the potential integration of Carbonara with other cutting-edge technologies. By 2025, we can expect to see AI models evolving beyond purely transformer-based architectures that have dominated the field. This evolution could lead to more sophisticated and efficient protein design capabilities, potentially enabling Carbonara to tackle even more complex molecular challenges. The intersection of AI and biotechnology is likely to yield groundbreaking advancements in drug discovery and personalized medicine. As consumer AI heats up, we may see the emergence of AI-driven applications that bridge the gap between consumer and enterprise use. In the context of Carbonara, this could manifest as user-friendly interfaces that allow researchers and even citizen scientists to engage in protein design, democratizing access to this powerful technology. Another frontier for Carbonara lies in addressing global challenges, particularly in the realm of climate change and sustainability. As the carbon credit market expands and evolves, there's potential for Carbonara to contribute to the development of novel carbon sequestration technologies. The model's ability to design thermostable enzymes could be leveraged to create more efficient catalysts for carbon capture and utilization processes, aligning with the growing demand for innovative climate solutions. The next frontier also involves pushing the boundaries of what's possible in materials science. Carbonara's unique approach to protein design could be adapted to create bio-inspired materials with unprecedented properties. This could lead to breakthroughs in fields such as nanotechnology, advanced materials engineering, and even space exploration, where custom-designed proteins could play a crucial role in developing materials suited for extreme environments. As we venture into this new frontier, ethical considerations and responsible innovation will be paramount. The scientific community's proactive approach in addressing potential risks associated with AI-driven protein design sets a strong foundation for future developments. Balancing the immense potential of carbonara with necessary safeguards will be crucial as we push the boundaries of what's possible in protein engineering and beyond. While carbonara represents a significant advancement in protein design, its implementation faces several challenges that researchers and developers must address. One of the primary obstacles is the complexity of protein folding and function, which makes it difficult to accurately predict and design proteins for specific purposes. Much like the intricacies of perfecting a carbonara recipe, where every small element from ingredients to preparation steps must be precisely right, protein design requires meticulous attention to numerous factors. The quality and completeness of input data present another significant challenge. Carbonara, like other computational models, relies heavily on existing protein structure databases. However, these databases may not encompass all possible protein structures and molecular interactions, potentially limiting the model's ability to design novel proteins for unexplored applications. This challenge is analogous to the difficulty in finding truly comprehensive and accurate carbonara recipes online, where even the best sources may be incomplete or slightly misleading. Another hurdle in implementing carbonara is the computational cost associated with processing complex protein structures and molecular environments. As the model deals with increasingly intricate designs, the demand for computational resources grows, potentially limiting its accessibility to researchers without access to high-performance computing infrastructure. This challenge is reminiscent of the specialized equipment and techniques required for perfecting carbonara, such as using a double boiler to achieve the ideal egg and cheese consistency. The black box nature of deep learning models, like carbonara, also poses a challenge in implementation. Understanding the precise reasoning behind the model's predictions can be difficult, hindering the ability to interpret and validate results. This lack of transparency can make it challenging to identify potential biases or limitations in the model's training data or architecture, similar to how the nuances of perfecting carbonara can be difficult to articulate and replicate without extensive experience. Lastly, the integration of carbonara into existing research workflows and its adoption by the scientific community present practical challenges. Researchers may need to adapt their methodologies and learn new skills to effectively utilize the tool. Moreover, as with any new technology, there may be resistance to change or skepticism about its reliability, requiring extensive validation and demonstration of its benefits before widespread adoption can occur. This mirrors the challenge of convincing traditional cooks to adopt new techniques or tools in their well-established recipes. The long-term vision for carbonara extends far beyond its current capabilities, promising to revolutionize multiple fields through its innovative approach to protein design. As we look towards the future, carbonara's potential to transform biotechnology, medicine, and environmental science becomes increasingly apparent. In the realm of biotechnology, carbonara's unique ability to consider complex molecular environments in protein design opens up unprecedented possibilities. The model's flexibility in handling various molecular entities, including nucleic acids, lipids, ions, and small ligands, positions it as a powerful tool for designing novel protein-based materials and therapeutics. This capability could lead to the development of highly specialized proteins tailored for specific industrial processes or medical applications, potentially catalyzing breakthroughs in areas such as enzyme engineering and drug discovery. The medical field stands to benefit significantly from carbonara's long-term development. As the model continues to evolve, it could play a crucial role in personalized medicine by enabling the design of proteins that interact precisely with an individual's unique molecular landscape. This could lead to more effective and targeted therapies, particularly in complex diseases where the molecular context is critical. Furthermore, Carbonara's ability to optimize sequence predictions based on specific constraints could accelerate the development of new vaccines and biotherapeutics, potentially revolutionizing our approach to treating and preventing diseases. Environmental applications represent another exciting frontier for Carbonara's long-term vision. The model's potential in designing proteins for carbon sequestration aligns with urgent global efforts to combat climate change. Recent research has demonstrated that custom-designed proteins can drive the formation of carbon-rich minerals, offering a potential pathway for enhanced carbon storage. Carbonara's context-aware approach could be instrumental in optimizing such proteins for maximum efficiency in various environmental conditions, potentially leading to scalable solutions for carbon dioxide removal. As computational power continues to increase and our understanding of protein-environment interactions deepens, Carbonara is poised to become an even more powerful tool. The long-term vision includes the integration of Carbonara with other cutting-edge technologies, such as molecular dynamic simulations and advanced sampling techniques, to create a comprehensive platform for protein engineering. This integration could enable the design of proteins with unprecedented levels of functionality and stability, pushing the boundaries of what's possible in synthetic biology and materials science. Ultimately, the long-term vision for Carbonara is to serve as a cornerstone technology in the ongoing revolution of protein engineering. By enabling the design of proteins that can interact precisely with complex molecular environments, Carbonara has the potential to drive innovations across multiple industries, from developing new sustainable materials to creating advanced medical treatments. As we continue to explore and expand the capabilities of this powerful tool, we move closer to a future where custom-designed proteins play a central role in solving some of humanity's most pressing challenges.